Hello, my name is Emily Packer, and I'm a current member of the Andover High School Bodybuilder Club. My project explores revolutionizing MRSA treatment through CRISPR-mediated disruption of the VanS VanR system in order to overcome vancomycin resistance. Methicillin-resistant Staphylococcus aureus, or more commonly known as MRSA, has emerged as a significant public health threat due to its ability to resist multiple antibiotics, including previously effective ones like methicillin and penicillin. This resistance poses challenges in treating a wide range of infections caused by MRSA, from skin abscesses to life-threatening conditions like sepsis. These are both within healthcare facilities as well as community settings. Moreover, the evolution of MRSA strains that are resistant to last resort antibiotics like vancomycin further complicates treatment options and raises an alarm within the medical community. Vancomycin plays a pivotal role in the treatment of MRSA infections by targeting the bacterial cell wall synthesis mechanism. This drug disrupts a critical step known as peptidoglycan synthesis, which is vital for the structural integrity of bacterial cell walls. Vancomycin's mechanism of action involves binding to the D-ala terminus of peptidoglycan precursors, essential building blocks required for cell wall formation. However, as previously stated, MRSA is starting to develop a resistance to vancomycin. Current treatment protocols often include combining vancomycin with other antibiotics or with beta-lactamase inhibitors to enhance efficacy. However, this approach has limitations, particularly regarding the potential of MRSA to develop resistance to these combination therapies. This highlights the urgent need to explore alternative strategies to combat antibiotic-resistant MRSA strains effectively, particularly targeting the resistance mechanism directly. Therefore, gene editing interventions could be explored. The mechanism for vancomycin resistance is called the VanS and VanR system, which includes a protein interaction that turns on several vancomycin resistance genes. The VanS protein acts as a transmembrane sensor histidine kinase. It detects vancomycin in the environment. Upon binding to the drug, VanS undergoes autophosphorylation, subsequently phosphorylating VanR, a response regulator protein. This phosphorylated VanR acts as a transcription factor, initiating the expression of critical vancomycin resistance genes like VanA, VanX, and VanH, as well as many others. These genes play critical roles in vancomycin resistance. For example, the VanA gene encodes a ligase enzyme responsible for modifying peptidoglycan precursors. This modification alters the binding sites for vancomycin, rendering the antibiotic ineffective. Additionally, the VanX protein gene hydrolyzes the D-ala dipeptide, and the VanH gene participates in the biosynthesis of altered peptidoglycan precursors. To counter resistance mechanisms in MRSA, a specialized biological system harnessing the CRISPR-Cas9 technology has been developed. This system employs guide RNAs to steer the Cas9 enzyme to specific locations in the genome, enabling precise modifications. The primary focus of this intervention is on inactivating the VanS and VanR genes, crucial components of MRSA's vancomycin resistance mechanism. Targeting these genes aims to disrupt the activation of other vancomycin resistance genes downstream, essentially shutting down the mechanism responsible for vancomycin resistance. Key components of this system involved guide RNAs that target both VanS and VanR, as well as proteins, terminators, and the Cas9 enzyme. The gRNA serves as a critical component, guiding the Cas9 to the VanS gene. The VanS gene gRNA directs the Cas9 precisely to the VanS gene, inducing breaks in the DNA at specific locations. This disruption hampers the sensor's kinase function, impeding its ability to activate the vancomycin resistance pathway. Similarly, the VanR gRNA targets the VanR gene. By guiding Cas9 to VanR, the VanR gRNA causes breaks within the VanR gene, disrupting its regulatory role. This disruption prevents the activation of downstream resistance genes such as the VanA, VanH, and VanX genes, and when exposed to vancomycin, MRSA will become more sensitive to it. So, in essence, the precision of the Cas9 in targeting the VanS and VanR genes offers a promising avenue to combat versus resistance to vancomycin, potentially reshaping the landscape of antibiotic treatment and challenging these infections.